from the very, very beginning when I was still a PhD student. And so I was lucky enough that I was given the opportunity to really follow my passion and to really follow, let's say, my really inspiration from a certain point of view. So, I mean, I consider myself very, very lucky. So as probably you know, but if you don't know, basically I'm a food microbiologist. So my talk today is going to be about food microbiology, but on a specific subject that is actually the link between the food and health as modulated by the human microbiome. So I just want to give or to have a confirmation that you are actually seeing my screen at this moment. Yes. Okay.
Yes.
Great. Great. Thank you. So as you can see, there are going to be several aspects that, oops, sorry, I did something. Okay. There are going to be several aspects that are going to be included in my presentation, but really, the central point here is the role that the microbiome has in the modulation and in the influence, basically, of the human health. Just for logistics, if you have questions, I would suggest that you put questions in the chat, and then we can have all the questions taken at the end. And of course, if at the end you would like to pose your question vocally, even better, so that we can have a little bit of live discussion at the end of the talk. When we're talking about microbiome, and especially human microbiome, and even better, gut microbiome, because gut microbiome or intestinal microbiome was one of the first topics in the frame, this big, large frame of microbiome studies that was really investigated and really addressed. There has been a lot of interest in the scientific world at the beginning, and this is witnessed, let's say, by the covers of very important journals, top journals in science, that gave to the microbiome topic the space in the cover. So, Nature, Science, Cell, all those very, very important journals have dedicated time or dedicated space actually for this topic, because in the last, I would say, 20 years, really microbiome science has grown a lot. And if I have to put microbiome science in the different context of the food system, of course, when it comes to human nutrition and human health, still microbiome science probably is at the top. For me, this is a little bit, let's say, I'm not saying that is upsetting, but it tells me that as a food scientists, we need to push for microbiome science also outside of the nutrition and health, because in the food system there are a lot of spaces where microbiome science and microbiome application can really represent a driver for innovation and a driver for change. Um, it does make sense that microbiome science was first applied to humans and to human health, because if you consider the way we are built in terms of, let's say, the way that we are constructed and our body is constructed, we have a lot of different districts that can be highly colonized by microbes. And actually, me, I always like to consider ourselves like a super host, or in a more, let's say, vulgar terms, a container, because we are containing a lot of different microorganisms in different organs, and especially those that have some kind of access to the outside world, considering with respect to our body, basically. So here you see all the respiratory tract and of course the gastrointestinal tract, but also the urinary tract, especially when it comes to the female reproductive organ, let's say. So those are the places where you have the biggest concentration of microorganisms, but let's not also forget about skin, for instance. And nowadays there is really a lot of push towards skin microbiome, because we have the possibility to find solutions also to cure or to prevent some of the skin diseases that are at the moment relevant. So we have this great opportunity when it comes to human health, and of course gut has been the central, let's say, the central interest in the last 25 years, because in the intestine, first of all, we have a huge population of microorganisms. And so you can see here a number. We can have up to 10 to the 12 colony forming units per gram of feces. And so if you consider the amount of feces that we may have in our intestine, and you do the calculations on how many microbial cells we may have in our intestine, those can overcome the total number of the cells of which we as humans we are made of. So there is a huge amount of cells in our intestine with a lot of diversity. You see some numbers here. But what I would like to also say is that the intestine is also a super important site in our body for immunological response. So we have immunological sites that can be stimulated, and as a consequence can somehow enhance the ability of our body to defend itself against pathogenic microorganisms or external I would say elements, because it can be also other things that are entering, and they can toxins, for instance, that can be combated. But the intestine, and let's not forget about it, is also a very important site for neurological stimulation. So in the intestine, there are peripheral neurons that are directly in connection with the human brain. And as a consequence, we should not forget about the possibility to stimulate uh, our brain with things that are happening inside of our intestine. And this has opened up like a huge area of research, because for instance we are not anymore focused on diseases that are still non-communicable, but they are really located at the level of the or at the level of the intestine, or the main manifestations are at the level of the intestine. Think about gluten intolerance, or think about some kind of again also intestinal cancer, for instance. But we can also think about other diseases that are far away from the intestine, like Alzheimer, Parkinson, depression, all these kind of things. So again, we have a huge potential in study the microbiome. And here, if you see at your right, I have just mentioned two words that is important to have a kind of clear indication of what they are, because they are not synonymous, and so we cannot use one instead of one in the place of the other, because they have completely different meanings. So when we're talking about microbiota, we are basically focusing on the ecological setup, ecology. Okay, so we are constructing a list of the microorganisms that are present in a specific ecosystem. If we want to study, or if we consider also the activities and the functions that those microorganisms are expressing, then we are making more complex the experimental scenario, and we are switching from the study of the microbiota to the study of the microbiome. So microbiome is something that takes into consideration ecology, but is implementing the ecological information with the functional information, metabolism, byproducts, metabolism, all these kind of things that may be extremely, extremely important when it comes to digestion, when it comes to absorption, when it comes to production of specific compounds that can be useful for or beneficial for human health and all these kind of things. In this slide here, you have a very complex illustration of what the gut microbiota is responsible for. And so on top, you have some, I would say, influencing factors that are really modulating the gut microbiota and as a consequence, gut microbiome. Very, very important, the delivery mode. So it is already very well accepted that C-sections deliver babies that have a completely different gut microbiota when compared to babies that have been delivered naturally, let's say. And this is not only at the moment of the birth and immediately after the birth, but is also respected, or say is also confirmed in more older ages, let's say. So it seems that again this is very, very important in order to establish the gut microbiota of one individual. Of course, genetics, age, lifestyle, like exercise, diet, and here is something that we are going to be focusing on, but also stress, and of course, antibiotics can have a huge, huge impact on the diversity and function of the gut microbiota. And around you see some of the, let's say, some of the actions or functions where gut microbiota is important. I'm not going to go into detail because we don't have the time, but for instance, the integrity of the epithelial barrier. Microbiota helps to keep those cells very tied together so that there are no spaces between the different cells, and as a consequence, microorganisms, for instance, cannot penetrate. Imagine a penetration of a pathogenic microorganism that can then be translocated. Of course, microorganisms in the gut, especially in the colon, they are responsible for fermentations, and so they degrade the big molecules, especially those that humans have not been able to digest, like fibers, for instance, into smaller molecules, and those molecules can be absorbed and can have beneficial effect on human health, both from the side of the digestion itself, but also for the side of the function, specific function that this molecule can have. Stimulation of the immune system. Again, metabolism. We know, for instance, that fibers are metabolized for the production of short-chain fatty acids. We are going to be listening about SCFC because those are very, very important physiological for humans. Protection, we said it, but also drug metabolism. So we should not forget about the fact that when we are assuming a medicine or a drug, this drug may be undergoing a transformation in our gut because of the gut microbiota. So this is something that has to be considered, because if the drug is transforming something that from active is not becoming active, you can understand that the drug is not anymore efficient, or in any case does not have efficacy. But it's also important to take into consideration the effect that the drug has on our gut microbiota, because this is the example, the most stupid example is the one about antibiotics. And so, you are, yes, getting the antibiotic because you have a pathogenic agent that has to be combated. But if this antibiotic is swiping away all the microbiota that you have in your gut, this is actually something that is not good. And the other thing that I would like to underline before getting a little bit more concrete is that the gut microbiota, and in particular the biodiversity of gut microbiota, is extremely, extremely important, in a sense that the higher the biodiversity, the lower the risk for diseases. And so if we stay here with a bacterial gene count that is high, meaning that the biodiversity is high, you can see that there are a number of different parameters that can benefit human health, from decrease of gut permeability, decrease of endotoxemia, pro-inflammatory cytokines are decreased, for instance, production of beneficial active molecules, all those activities, let's say, or all those parameters that again are positive for human health. And as you can see here, these type of manifestations are actually pushed or prone by healthy diets, healthy lifestyle, use of fibers, prebiotics, probiotics, but also fecal transplantation, if the fecal transplant, and considering fecal transplantation a kind of, you know, extreme treatment, because at the moment fecal transplantation is really something that is applied clinically as a cure for quite heavy diseases that cannot be treated otherwise. Okay, and one example, classical example, is infection with Clostridium difficile, for instance, that hardly can be addressed with another strategy, basically. If the diversity is decreasing, then you go into dysbiosis. And dysbiosis means a lot of different increase of detrimental or non-beneficial effects. And as a consequence, you can have non-communicable diseases, and as you can see here, diabetes, cardiovascular disease, inflammatory bowel disease, you can have also cancer, etc., etc. And this is actually pushed or influenced also again by the diet, because what we call the Western diet, that is quite rich in fats and sugars and proteins, that can be, and animal proteins, that can be, let's say, increasing the risk of getting sick with this kind of disease. How can we modulate gut microbiota and gut microbiome? I just want to show you a couple of studies that we have been running in the last 10 to 15 years, where we are really demonstrating how diet is an important element that can be considered for increasing diversity and modulating the diversity. The first study that I would like to show is about the differentiation between the gut microbiota in omnivores, vegetarians, and vegan subjects. So this is the result of a study that we have run back in I was in a project in 2015, 2016. Even better, even before, sorry, because this study has already been published in 2015, so probably the project was earlier than that. But here, what I want to show is that every single spot that you see in the graph is representing a subject that we have recruited. We have studied from the point of view of the dietary diaries, so understanding exactly what these people are eating. And then we got a fecal sample and we ran ecology studies. So basically what we did was to sequence the populations that they had in the feces, and this specific spot represents the summary of the species, let's say, that they have, is representing basically the ecology, the intestinal ecology. The reds are the omnivores, the blue are the vegetarians, the green are the vegans, and as you can see in the plot, we cannot really differentiate the subject. So what I just said seems to go exactly opposite to what I was telling you before when we said that diet is a very big, or let's say, is a good modulator of the gut microbiota. Because again, here we didn't see really a difference. I just want to point out that this was done with a quite low efficient methodology for ecology studies. This was based on an electrophoretic method where species were detected by bands that we know it was not able to go deep in the identification of the microorganisms. So I would say that having or using this technique, we could only have like a rough picture of the main microbial populations that were present in the feces of those subjects. Okay, so this could be a kind of limitation, and that's why what we have done was to use another approach to confirm the results that we got with this methodology by really looking at 16S rRNA gene sequencing. So this is a quite, let's say, efficient method with a quite deep level of, let's say, sensitivity also towards the detection and identification of the species. And here you have these two plots where in the first one, you have again subjects that are omnivore, vegetarian, vegans, sorry, and vegetarians. And here you have another type of clusterization that is based on the high, medium, and low adherence of the Mediterranean diets. Because what we have done in this study was to actually analyze the diaries that the subjects have produced by looking at the adherence towards the Mediterranean diet. We know that the Mediterranean diet is one of the healthiest diets in the world. So we were interested to understand if Mediterranean diet could be high only in vegans and vegetarians, or could also be high in other subjects. And actually, what you can see here is that, yes, the large proportion are vegans and vegetarians, but you have also a small proportion of omnivores that are highly adherent to the Mediterranean diet. So again, is not the type of diet that you're using that guarantees the fact that you are adherent or not adherent, because look at also here, you have these subjects here that are vegan and vegetarians but are medium adherent, and you have even a small proportion of vegetarians that are low adherent to the Mediterranean diet. So, I mean, is not really about the diet itself, is really about the type of food that you are eating. But again, here the bottom line is that no matter what we considered as a parameter for clusterization, the appertenance of the subjects to a specific category diet, or the level of adherence to the Mediterranean diet, again, there is no differentiation. So these subjects are actually quite homogeneous in terms of the populations that they have in their intestine. And here I'm stressing out once more that we're talking about ecology. So we are not talking about functions, we are talking about ecology. So comparing the list of microorganisms that these subjects have in their intestine, we could not really see any differences considering or based on the criteria that we have used for the clusterization. But something quite interesting was then pushed ahead in order to try to explain these evidences that we have obtained from the taxonomical and ecological point of view. Because when we moved forward towards taking into consideration also the functions that those microorganisms are actually expressing and they are responsible for in the intestine of those subjects, we could really see a difference. So here you have a PLCA of the results that we obtain with metagenomics. So metagenomics is the methodology that is the gold standard for the study of the functions of the microorganisms in a complex ecosystem by next generation sequencing. And so you can see that at the moment we have the results of the metagenomics. I mean, omnivores, vegans, and vegetarians are quite differentiated. And this differentiation is pushed in premise by the carbohydrate and protein metabolism genes. So omnivores are enriched with those genes with respect to the other populations. But what we have seen also, and here you see in this dendrogram, that in omnivores, you also have a decrease of certain genes that are responsible for mobility of the microorganism. So most of the genes that you see here are actually encoding for flagellas in the microorganisms. So we don't really know exactly what is the physiological meaning of decreasing or increasing the genes encoding for flagellas, but flagellas in the last years have been considered as important microbial components that can also be used in delivery, for instance, of drugs, but also in the immune treatment of cancers, for instance. So again, we don't have a definitive answer towards the significance or the physiological significance of the picture that we see here, of this trend that we see here, but again, there is a quite big modulation of what the microorganism can or cannot do. And this was also confirmed if we apply metaproteomics. So as you can see here, we are moving from DNA to proteins. So by looking at the metaproteomic profile of the omnivores, vegans, and vegetarians, again, we can see them very, very different. So they are differentiated. Quite interesting is the fact that in the vegans and vegetarians, we have an overpresence of genes that are responsible for the production of short-chain fatty acids. And so, again, it is somehow confirmed that if you are eating a lot of fruits and vegetables, you are feeding your microbiota in the intestine with fibers that are the primary source of short-chain fatty acid production. So microorganisms, they are using the fibers for the production of those compounds. But it was also quite interesting by, I mean, looking at the production of the genes responsible for the production of the vitamin of the group B. And here you see that again, vegans and vegetarians had a overrepresentation, while the omnivores did not really have genes responsible for that. Consider that vitamins of the group B are mainly coming from meat products. So bean meat present in the meat, microorganisms are actually acquiring, and we are acquiring vitamin B from food. When meat is missing, and this is the case of both vegetarians, but even more vegans, because in vegetarians you may have some kind of animal origin foods, in vegans not. You can see that as if the microorganisms are supplementing with the production of vitamins B, with the group B vitamins, because they are actually lacking in the food that we are eating. So by this point of view, it's very interesting to to see here a kind of collaboration with the microorganisms that the vegans and vegetarians have in their gut, because they are actually helping the subjects with the supplementation of vitamins that are missing in their foods. And the last thing that I want to show as a kind of modulation, let's say, of the gut microbiome and microbiota, is the fact that if we in the diversity of one specific species that is commonly found in the intestine, that is Prevotella copri, we could really see a difference in the strains of Prevotella copri that could be identified in the intestine of the omnivores and in the intestine of the vegetarians and vegans. So here you see that, so we put together vegans and vegetarians here. So they are actually different from the strains that we are reconstructing or we are detecting in the case of the omnivore. So the species is the same, but the strains are actually different. And you can see here also here that those that are in red are coming from the omnivores. The ones that are in green and in fluo green, they are coming from the vegans and vegetarians. So diet at the end of the day probably is not modulating the diversity in terms of species content, is of course modulating the activity, but is also influencing the strain composition within the species. So it may be that we didn't see any difference in the species level because they are all the same species here. But if we go a little bit deeper in the characterization or in the study of the specific species or the specific strain, sorry, we can see differences that are really guided or driven by the diet. Okay, so this was a little bit the introductory part, just to make you realize how important is diet, let's say, in the modulation of our gastrointestinal tract, and of course diet is something that and food in general, I mean, is something that is very important for us because we are getting nutrients and we are getting energy from the diet. So the first, let's say, the first question then diet is answering to is really the need of, or the necessity, not question, I mean the necessity the diet is addressing is the need that we have to get nutrients. And this is really happening with the digestion and update of food components. But let's not forget about the fact that the diet has components and have some nutrients or some compounds that us as humans, we are not able to attack or we are not able to digest simply because we are missing the enzymes that are responsible for the degradation of such compounds. And those compounds are actually reaching the intestinal environment where there are microorganisms which can promote fermentations. And those fermentations are able to produce a lot of different metabolites. Some of them are quite well known. Again, short chain fatty acids, for instance, are very well known for also for their beneficial effect on human health. But there are also producing a lot of other compounds that can be considered really as a messenger. So probably you have heard about the gut-brain axis, right? So the gut-brain axis is the concept of um, putting in communication the what is happening within the intestine with the brain through the exchange of stimuli that are mainly coming from chemical messengers. And those chemical messengers usually or often are metabolic compounds that are produced from the metabolism of the microorganism. So again, it's quite important to have a quite open mind regarding the interactions that are taking place in our body, because there may be something happening for the good and for the bad, something in our intestine that then is translocated around the body and is targeting several different organs. And actually, in the last years now, we are not anymore only talking about um, uh, gut and brain, but we are talking also about gut and liver. We are talking about gut and kidneys. We are talking about a lot of different connections that we are slowly realizing while science is advancing. And of course, as I said before, since we are like a superorganism in which us as humans we are hosting a lot of different microorganisms, there is a very intimate interrelationship between the microbes and us as humans, because we can influence the microbiome diversity function with compounds that we are also producing, but most of the times that we are assuming or we are ingesting or we are having, because again, drugs is something that can also come from different ways. But we need also to take into consideration that there are some compounds from produced by the metabolic activity of the microorganism, but not only, because there are going to be also interactions that are taking place very, or let's say at the level of the surface of the epithelium of the intestine. And those are the ones that are actually also promoting some kind of beneficial or non-beneficial effects on human health. So there is going to be also, again, the counterpart of the influence that the microbiome itself can have on human health. And there are going to be already, or there are already some very well established effects regarding the impact, let's say, of the microbiome. Of course, when we are talking about food and the modulation of the microbiome, I mean, the most simple and common way of influencing the microbiota of the intestine is really about probiotics and prebiotics. Probably you know exactly what this means, but I just wanted to show it as a kind of reminder for us that those probably are the most effective ways we can modulate the microbial ecology, either by introducing live microbes in sufficiently high quantity that can modulate the ecology. And so when we're talking about probiotics, we are really talking about live microorganisms. This is a microorganism that has been selected and that is actually ingested in high quantities. Usually, probiotics are not present in high quantities in food generally, unless they are added. So you can have a food that has probiotic activities, but the food has probiotic activities only if you are adding a high quantity of a non-probiotic inside. Okay, and this is something that I'm going to be coming back at some point. And there are also prebiotics. So the prebiotics are not living cells; they are components. There are let's say chemicals that are present in the food, and substances that are present in the food. Don't like a lot the word chemicals. Let's put substances that are present in the food that can really serve as a feed for the microbes, or as a food for the microbes, call it the way you want it, because of the ability of the prebiotics to enhance the growth of probiotics or the growth of beneficial microorganisms. You can understand that if we are eating foods that contain prebiotics, we are actually pushing or enhancing the capability of beneficial microorganisms to grow inside of our intestine and as a consequence have positive impacts on human health. But when we are talking about microbes in foods, we don't only have probiotics. We also have foods that are containing a high quantity of microorganisms because the microorganisms are the responsible for their production. When we consider foods and microbes, there is a quite nice categorization of the foods based on the estimation of the amount of living microorganisms that they contain. And so you can see here that, you have foods with a quite low quantity of microorganisms. Here it is indicated below 10 to the fourth. Then you have microorganisms that are present a little bit higher, up to 10 to the seventh, in a medium contaminated or in a medium, let's say, load of microorganisms. And then you have the high, so that are the ones above 10 to the seventh. Foods with low load are primary pasteurized. The ones that are medium are the ones that are eaten raw, like for instance, fruit and vegetables, for instance. The ones that are high are the ones that either have been fermented or they are added with specific microorganisms like the probiotic. And so here I would like to spend like a couple of minutes on this important role of fermented foods, because fermented foods can be really considered as a kind of good supplementation of microorganisms to our intestine. And the microorganisms that are responsible for the fermentation, I would not call them probiotics because it is not correct to call them probiotics, but for sure I can call them beneficial because they are actually exerting some activities that are quite important for us as humans. And you can see how important is the fermentation as a metabolic activity that is happening. So first of all, fermentation is a process that is really able to valorize, I would say, the nutritional components that are present in raw materials. Raw materials like milk, meat, for instance, are extremely deperishable. So they don't have a long shelf life. But at the moment they are fermented, and of course, when we are talking about fermented foods, we don't have only to consider the fact that fermentation is happening. During fermentation, and especially after fermentation and during maturation, you have a lot of chemical-physical changes that are happening. Mainly, you have loss of water, and as a consequence, you have lower water activities in your food. So fermented foods for sure are much stable foods when compared to the raw materials. And since during the fermentation there is acid that is produced, they are also much safer than the raw materials. So, for instance, raw milk may not be safe, but if this raw milk is used for the production of a long maturing cheese, like Parmigiano Reggiano, Grana Padano, Gouda, all these kind of cheeses, the cheese is absolutely safe because you have all these kind of hurdles that are introduced that are blocking the development of the foodborne pathogens. What is the functions that can be more relevant for human health? For sure, there is all let's say the group of the metabolites that are produced by the fermentation. So we said already short chain fatty acids, but also other compounds like, for instance, some metabolic products of the degradation of polyphenols, for instance. that are quite important for a number of different aspects, let's say, in the human health. We should consider fermentation as a kind of pre-digestion of the food. So when we are eating fermented foods, we are eating foods that have already a low level of complexity from the chemical point of view with respect to foods that have not been undergoing a fermentation. And this is because there are enzymatic activities that microorganisms are putting in place in order to get nutrients out of the food in a way that they can be sustained from the nutritional point of view. Okay, so again, fermented foods is a topic that has acquired a lot of attention. And when we are talking about fermented foods, here it comes what I was telling you before. We need to be very careful in the definition or in the correct definitions, because as you can see here, there are some definitions that have to be respected. So when we are talking about probiotics, here there are live microorganisms with certified health benefits to the host. Okay, so probiotic is a microorganism for which beneficial actions have been identified and have been proved. And usually this proof is through clinical trials. Okay, so usually, and especially Europe, is very strict on the definition of probiotics. So probiotic is really a microorganism for which has been proved the benefit for human health with a clinical trial. On the other hand, you have fermented foods, and then you have these probiotic fermented foods. So when you are talking about fermented foods cannot be considered probiotic if we do not demonstrate that they contain probiotics, or more simply, they can be called probiotic fermented foods if we are adding probiotics in the fermented food. Usually, we cannot define a fermented food as probiotic if really we do not demonstrate that they contain probiotic microorganisms. Especially when a fermented food is obtained by spontaneous fermentation, so we are not inoculating anything, but is a spontaneous fermentation, those fermented foods, not at all, they can be considered probiotics. Okay. Also because it could be very complicated to demonstrate that they contain microorganisms, that they are probiotics. As I said before, we can call them beneficial, but we cannot call them probiotics. Okay. And as a last part of my talk, I'm going to go very, very fast also because we are getting close to the end of the class. I just want to show you two strategies that we have used in order to really try to modulate the gut microbiota and gut microbiome in humans, because actually the first study here is in vitro using a simulator of the gastrointestinal tract of humans, that is the SHIME, and the second one that is really about humans. So the SHIME, as you can see here, is basically a long let's say a group of fermenters, are like a lot of vats that are connected together, and they represent different parts or different districts of the human intestine. And those vats are actually connected through all these pumps in a way that we can also simulate the movement of the intestine. We can transfer from one district to the other one, and so we can really run quite, let's say, controlled digestions or fermentations as it happens inside of the human gut. What we have done was to develop what we called superfood, that is really taking into consideration the main characteristics of the Mediterranean diet. So here you see source of phenolic compounds, fatty acids and phenolic compounds. Again, broccoli and moringa, walnut. I mean, a lot of different healthy ingredients to produce this superfood that was then tested in this gastrointestinal simulator. And here you see a little bit the experimental approach that we have used. And so we first have 3 days of stabilization of the system. So we need to stabilize the inoculum that we are using, because we are always using feces as the inoculant for the chyme. We need this inoculum to stabilize in order to get adapted to the medium where the fermentation is taking place. And after 3 days, we have fed the inoculant with the superfood or with a placebo. Okay, and this was done for 1 week. So every day, twice per day, the chyme was fed with this superfood, and after 7 days, we stopped this supplementation either of the placebo or the superfood, and we had 3 days of washout, meaning that we did not add the superfood or the placebo, but we went ahead with the fermentation just using the synthetic medium to feed the microorganisms inside of our vats. And what we have seen was quite interesting from the side of the metabolic activity. So here you have the results of the short chain fatty acids and the volatilome. What I want to point out here is if we take into consideration the different districts of the intestine, so the ascending, the transverse, and the descending, you can see that from the treatment 1 and 2, the treatment 1 is the one with the Mediterranean diet based food, and the other one is with the placebo. You can really see an increase of the specific short chain fatty acid, acetic, propionic, butyric. So you can see that there are differences and significant differences in the quantity. So you have higher concentration of short chain fatty acids when you are using the superfood. And I would say that this is also remaining active when you have the washout. So if you see always the parameters here of the difference between the treated and the placebo, you can still see a difference. So there is a kind of remaining effect that is present inside of the vats. I mean, we're talking about vats here, because still the microorganisms are somehow producing short chain fatty acids, even if we don't have anymore the superfood. So we can really recognize a kind of a modulation that is also stable, because this is something that you see here, because those are all clusters of these samples before, during the adaptation. So here is before feeding with the Mediterranean diet based food. Here is with the placebo. So already here you can see the difference. And then of course, when we have the treatment, look at the treatment with the Mediterranean diet based food and the treatment with the placebo. So completely different, separate situations. That means that the microorganisms are actually behaving completely different. Probably there is a difference in the ecology, but for sure there is also difference in the function. And this aspect is actually underlined here with the microbiota profile. So here we didn't do really microbiome, but we did microbiota. You can see that the ecology here is completely different. So first is the placebo, then you have the superfood, so very, very different. And if we dig down in the species, or actually in the genera of the microorganisms, you can see, for instance, an increase of beneficial microorganisms like Faecalibacterium or Bifidobacterium, and a decrease of other ones that have been described as detrimental, like for instance the Klebsiella or Sutterella. And the last slide that I want to show you is about the probiotic story. I call it probiotic story because I think that probiotics is probably one of the best business models for companies, because if you are a probiotic company and you are selling a probiotic, at the moment you are able to demonstrate to the consumer that this probiotic is very useful for your health, you will always have to buy the probiotic and consume the probiotic to keep this benefit stable for your health. If you are stopping, no matter what, it will disappear. So probiotic, as I said before, is very, very good strategy from the business point of view. Here, what we have done was to monitor the modulation of the gut microbiota of a number of subjects that were consuming a probiotic yogurt. So we had in our collection strains that of Lactobacillus rhamnosus that were actually characterized by their potential probiotic activity. We have designed this kind of intervention in which we had the inversion, let's say, of the group. So at the beginning, group one was getting the probiotic and then was getting the placebo, while the group two was getting first the placebo and then the probiotic, the putative probiotic. And here down, now it is very small and probably also in your screens is small, you are not able to see it, but we have a decrease of populations like Bacteroides, for instance, that are negative, and the increase of other microorganisms like this one that is Prevotella. We talked about it before. Okay, so for sure here we see already a quantitative modulation because there are populations that are growing and populations that are decreasing. A partial overlap between the two groups. So you can see here that we have some species that we have also seen here increasing, like Faecalibacterium and Prevotella, that are actually driving the differentiation. But what is most important, in my opinion, is looking at the overall spread of those points that you see in this graph. So those points are really representing this red, the baseline. Then you have the blue, that is the probiotic intervention, and then you have the yellow, that are the ones of the placebo intervention. Okay, so you can immediately see how the ecology is changing, because here again we are talking about ecology. So the moment the probiotic is subministered or is consumed, the ecology is changing a lot. And then, based on the different subjects, this differentiation is either maintained at the end of the period, because, for instance, in this case, you have the 0 here, the 42 days here. So this, let's say that the ecology here is getting back to what it was, differently from this group with this subject here. This is the subject one, in which the sample at 0 was here, and then through the intervention we had a quite big move of the ecology, reaching after the washout this point here. So here it was really, let's say that the supplementation and the consumption of the yogurt really had an effect on differentiating and maintaining a different ecology following the yogurt or the probiotic supplementation. This actually gives me the possibility to get to the last concept. When we are addressing gut microbiota, gut microbiome, as it is a little bit described by these graphs here, by these plots here, we have to deal with a very, very large intraspecies diversity. As humans, we are very diverse. And we are diverse not only because we have different backgrounds, we are coming from different regions, we are different because of the genetic setup that each of us has, but we are also very different from the content of the microorganisms in our intestine in terms of ecology. So working with gut microbiome in healthy populations is very, very complex, because you really need to be careful about the outcomes that you want to see. Because if it is not clear and you say, I'm going to be doing something in healthy populations and see what is happening, forget about it. You're going to be getting lost. So very, very carefully identify the question that you want to address. The complexity is getting a little bit more controlled at the moment you switch from healthy populations to diseased population. And this is exactly connecting to what I was telling you before, that when you are switching from health to disease, the microbial diversity is decreasing. And so microbial diversity in diseased subjects is lower, and as a consequence, you can easily see changes that are happening in the gut microbiota, gut microbiome, because the level of complexity is lower. Last concept, let's not forget about safety. I mean, this is a correspondence that we have published several years ago when this big explosion of the interest around fermented foods was taking place and still is very high. Fermented foods, they can be very, very good in terms of contributing with the taste, diversification. So really, they are a source of new, I would say, landscapes when it comes to the food system. However, we need to be very careful about the fact that they have to be safe. So safety of fermented foods is really, really guaranteed when they are produced in the appropriate settings, or let's say production settings. I'm not sure what is happening when every single consumer is fermenting foods in their own kitchens. I mean, nowadays there is a lot of interest around plant-based foods and vegetables fermented at home. Well, let's be careful, because if we are not respecting the right conditions for fermentation, we may have foodborne pathogens that are remaining inside of our foods, and they represent a risk for our health. Good. Thank you very much. I'm going to be stopping the sharing, and I'm going to be totally open for discussion and questions. I mean, I don't see any questions in the chat because I believe that you will be happy to open your microphone and put some questions or comments live. So really looking forward to your contribution at this point. Any aspects that you would like to address a little bit more specifically? Not really. I don't see any kind of any kind of reaction from your side.
I have a question.
Sure, go ahead. Good.
This is regarding a bit of how we can actually promote this um, practice towards a wide range of consumers. So, preferably, what role do you see for prebiotics, probiotics, and fermented foods in the future, maybe in transforming industrial agriculture and food processing towards more regenerative
Mm-hmm.
and health promoting practices?
Yeah.
How do you think?
So this is a quite interesting topic that at the moment is maybe it is not addressed to the total potential that it may have, because we have been quite often used to think about processing itself and primary production itself, so without no real connection. So there are kind of requests or expectations from the primary production, and then also from the processing, that often are not really much in alignment, because when it comes to processing to primary production, especially in the past, there was this extreme push to quantity, and as a consequence, intensification, and we still see the detrimental effects of intensification in certain parts of, I would not say only in certain parts of the world, but in the certain parts of the system itself. And then on the other side, there were the processors that they were getting the raw material, they were just processing, I would say throwing on the market foods without really um, strategically or not strategically, I would say, without really thinking well or analyzing well what would be the reaction of the consumer. So in the past, the consumer was really only someone that was going to the supermarket and was buying the food and was consuming the food. Now, and here now, it has been already like 10 to 15 years where really the approach of the food industry has changed pretty much towards consumers. So now consumers are really seen as an important link in the food system that has always to be, let's say, not interrogated, but consulted or, discussed with the consumers. And in this overall, let's say, scenario, what we are starting to see is the strict interconnection that is absolutely needed between the different segments of the food system that in the past were very much separated. This has a huge, huge potential because um, looking at soil health, animal health, and well being, we can really produce raw materials that have higher nutritional values, that have higher levels of safety, for instance. And so we may really have the opportunity nowadays to push for sustainable production that are really connected to healthy foods and as a consequence human health. um, there are already studies, for instance, in milk that if you have intensification of the animal production for the for the obtainance basically of the milk, they can probably milk can also contain inflammatory substances instead of let's say less intensive production of milk, leveraging the nutritional values of the greens or the plants that are also growing spontaneously, let's say in the fields, that can benefit the animal health and as a consequence also produce foods and milk that have a higher nutritional value. It is already demonstrated, for instance, that if you are using a kind of regenerative agriculture, fertility and nutritional content of soils is increasing. And as a consequence, plants that are growing on that soils will have a higher content of nutrients. Also fertility. I mean, fertility through regenerative agriculture is increasing in soils. So again, I very much appreciate your comment because we really need altogether try to link even stronger these two segments, let's say, or these two compartments of the food system, because we really have an opportunity here. And microbiota really plays a role in this kind of opportunities that we have. Absolutely.
Thank you, Professor. Probably one more question, if I
Oh,
may.
sure. You
Just
sure. Go ahead.
seeing how many other researches you've done, what do you think has been the most feasible research you've done so far in the probiotic field?
So, for sure, let's say that I mean, there has been a lot of scientific outcome that improved a lot the knowledge in the scientific field. Honestly speaking, if I see the business that was generated from those studies, unfortunately, I cannot say that there is a lot that has generated a lot of businesses. For sure, when we are talking about especially microbiome in the field of human health, Um, this helped to design specific treatments that are saving life at the moment. The fecal transplants that I was just touching for a second are really the only therapeutic possibility for subjects that are for humans or for individuals that are affected by the Clostridium difficile infection. There is no other treatment. So the fact that we understood how microorganisms can really help us at the intestinal level and how we can then transfer a healthy microbiome, fecal microbiome, to a diseased fecal microbiome and have the healthy taking over the diseased, this is actually really, really saving the lives of those subjects. If we get out of the human health, because as I said before, human health is for sure the space that has been investigated the most. But there are already some kind of evidence also in other parts of the food system, like, for instance, biofertilizers or let's say modulators to improve the resilience of the crops. We know that there are microorganisms that can improve the water retention at the level of the roots of the plant. So think about arid systems. And especially when it comes to less developed regions in the world, having plants that can actually grow with a less need of water input. This is of extremely, extremely important and is a humongous. Is humongous because we can really again take advantage of some of the interactions that are taking place between different actors, we'd say, because I'm considering the plants and the soil and the animals also important actors in the food system in a way that we can really improve. So again, a lot of opportunities. I can say that probably, just to close up, I can say that probably we still need to address an important hurdle that is a policy here, because unfortunately, there are a lot of barriers that are coming from the policy. And so, for instance, in the field of primary production and plant primary production, it is not clearly classified what it can be done and what it cannot be done. And then usually also laws or regulations often are a little bit complicated. Not sure if you are familiar with the novel food regulation in Europe. Novel food regulation in Europe, that is the one that is considering all these kind of new technologies like biomass fermentation, precision fermentation, culture foods, all these kind of things, is extremely, extremely heavy and complicated. I mean, as a company, I mean, I'm always saying you have to be very, very convinced about pushing your product that can be defined as a novel food into the market, because the roadmap to get certification from the EU is so complicated and so expensive that unfortunately it may kill food innovation.
I agree. Thank you, Professor.
Any other comment?
Uh, Sorry, just a quick one on
Yeah.
the regulatory end. You're talking of restriction, but I just wanted to know what you think are the major innovation areas. I saw you mention something to that effect, but in passing. But I need to grasp what are the major areas for possible innovation within the
Mm-hmm.
probiotic world
Mm-hmm. Mm-hmm. Mm-hmm.
from your
So
perspective. Yeah.
yeah, 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 yeah. You know what? I just want to make sure that we are on the same line here, because when we're talking about probiotics, I would like you to let's say go out from the classical definition of probiotics of microorganisms that are beneficial for human health, because when we're talking about probiotics, I think that if we have been understanding that probiotics are also useful for animals. And so probably the word probiotic can also be used for the story of the animals, also because the supplementation of probiotics in humans and animals is very similar. We need also to start to use the word probiotic for plants. So the word probiotic should somehow become more overarching in the different domains of the food system, because they should represent beneficial microorganisms that are increasing the health of the host, independently if this is a cow, this is human, this is a plant, or even the soil. Okay, so to me, the spaces for innovation are really those where we can leverage the activities of the microorganism to help reaching a specific objective. We have been talking about fertility. We have been talking about nutrient content. Of course, when we switch to animals, we have a huge problem still to be resolved with the safety of the animals. And safety in the animals is really connected to antibiotic use, for instance. So you can see how there are different streams that can be investigated. I mean, I know that probably you would expect from me a more precise answer saying, innovation is the use of microorganisms to do this or the use of microorganisms to do that. I'm not giving you this specific answer because I really believe that it really depends on the application that you have in mind. Because, for instance, do you want to produce ingredients for the food company? Yes, go in precision fermentation. Precision fermentation is a technology that is available nowadays. So you can make use of microorganisms first. You can make use of artificial intelligence for the optimization of the process. So a lot of different things that can be put together for maximizing the impact that you want to have. Okay. And again, the microbial world is so diverse that basically you have a never-ending opportunity to explore activities that you may identify as useful for your business somehow, or for your research, or for your development. Put it the way you want it. Okay?
Thank you very much. That is well noted.
Thank you. Good. Anyone else? No? Okay. So I think that I'm the one that should say goodbye to you today, because again, Marcin is not present as a project manager of the Master in Food System, but for sure it has been a great pleasure for me to have you here. I'm sure that our paths are going to be crossing. I mean, I see the faces of some of my students that we just finished the class a couple of days ago, so I'm looking for their finals and for their exams. Not sure if in your paths the University of Turin is going to be involved or not, but if not, I mean, for sure we're going to be meeting in the graduation conference. Now, probably talking about graduation conference still looks a little bit very far away, but it's not going to be. I'm telling you, it's not going to be. It's coming so fast that you will see that you're going to be waking up tomorrow, and again, you will have to travel in Leuven, where we're going to be hosting the conference. So Jamil is going to come in Turin. You're very welcome. So hopefully we are going to be meeting in person. And again, I take this opportunity to wish you the best and very nice and relaxed holidays that are coming, and looking forward to meet you. Okay. Thank you very much.
Okay, take
Bye
care.
Thank you,
Take care.
Professor.
Bye bye.
bye. Bye. Bye
Thank
bye.
you, Maribad. Bye.
Bye bye.